Shall I proceed with that? Dear all, greetings from School of Environmental Studies. Its Professor and Director Dr. V. Sivanandan Achari is here to greet you. Today in this 14th invited lecture, my pleasant duty is to chair the session of the invited talk by Dr. Kaustabh Kumar Maiti. Dr. Kaustabh Kumar Maiti is the principal scientist of CSIR-NIIST, Thiruvananthapuram, India. He has so many years of experience in various areas related to chemistry and pharmaceutical science. The major dedicated research areas of Dr. Kaustabh is targeted drug delivery system development, TDDS, diagnostic nanoparticle probes, multimodal theranostic nanoprobes based on SCRS fluorescence, MRI as diagnostic modalities, and PDT, PTT, and chemo and immunotherapy. Synthetic modification of bioactive natural products isolated from plants. With great pleasure, I will inform you that Dr. Kaustabh Kumar Maiti is an award winner for his excellent service. Award from Singapore Bioimaging Consortium, SBIC, A-Star Singapore in 2011. In various levels, his contribution is unique in the field of chemistry and medicine. Today, Dr. Kaustabh Kumar is interested in giving you an illuminating talk on recent advances in cancer nanotheranostics. On behalf of the organizers, I wholeheartedly invite you, sir, to illuminate us through your lecture navigation. Thank you.
Thank you, sir, for your kind words of introduction. Shall I share my screen first? Is it visible to all?
Yes, sir. It's visible, sir.
Yeah.
Yes, sir. It is okay. Fine.
Yeah. So, thank you for inviting me for this talk, and thank you all the organizers, especially Dr. Naibin, for inviting me in this platform to give an overview what we are doing at CSIR-NIIST and recent advances on nanotheranostics. It is a key word right now. Most of the people who are working on nanotechnology and nanotechnology-based medicine. And today I am going to give an overview which mostly helps the students community and to know about the subject and its importance in the ongoing research activities in their future careers. So I just give you very basic things, starting from what is nanotheranostics and how it implies in many therapeutic fields. And I'm emphasizing today on its cancer diagnosis and therapy. So all of we know that about the nanoparticles, its synthesis, how it is engineered. So the nanoparticles, when it comes in a nanoscale dimensions, it has the property, or it can afford both therapeutic and diagnostic roles, which is referred to as nanotheranostics. So the nanotheranostics is a combination of therapy as well as diagnosis in a single nanoconstruct. So what I'm going to give you the brief overview about the cancer nanotheranostics and its real-time importance for monitoring and intelligent fabrications for the treatment of the personalized medicine. So coming to again basic things about the nanotechnology, and which involves the materials in a nanoscale. So as all of you know, it is 1 by 1 billionth of a meter size of a human hair. So if you just look on the, just a minute. If you just look on the different materials as a bulk material starting from water to a tennis ball, you can see what is the dimension size of all this in this scale. So we are going to play a nanometer size particles which has tremendous potentials. I'm going to say you why this potential is and how it can implies. Just before that, to feel that the bulk material starting from water to small molecule, small synthetic molecule to big molecule like protein or virus, then again is one cellular component like a cancer cells, and then coming to a thing which is a material, any kind of materials. So if you scale up all these things in a nanoscale, you can feel what are their positions. So this is just a picture where I can show you when you see in your human eyes through electron microscope how a bulk material features in the nanoscale dimensions. So as I said, my talk is just a general overview. I just want to discuss about the nanotechnology and why this nanomaterials is so important to us. Because its physical and chemical properties we can change entirely compared to the bulk materials. And that gives a tremendous potential in diverse applications in the field of healthcare, in the field of in any kind of other areas of research interest. This is, I used to say this is actually a review published a couple of years back in Angochem. Just a very good insight about the nanomaterials, its what is features. A nanomaterial is generally considered a material within 100 nanometer in size, and what its important features in terms of its surface, its shape, and its materials. So if you can see, if you can modify, or if we can modify its surface in different kind of materials, or if we can use different kinds of shapes, or if we can conjugate different kinds of biomolecules or polymers or any kind of materials, then we can make a complete nanomaterials which have its application as far we wish to do. So just a example about see the bulk material and the nanomaterial, how we can tune its color. And that color also be tuned from the bulk versus nanomaterials. So while coming again to that in nanotechnology and its nanomaterials, then we need to have its chemical properties, its mechanical strength, and also its electrical and magnetic response, which definitely give a very good insight about the applications of these materials. And one of the important things of the nanomaterials we should know that when you tune the bulk material to a nanomaterials, then we definitely gain about its high surface compared to the volume. So surface to volume ratio is a very crucial features of the nanomaterials to get it and its applications. So as I want to focus about the nanomaterial applications towards in biomedicines. So I want to say that these nanomaterials, as I said in the previous slide, that its size and the nanomaterials which can ready to interact with the biomolecules is a very precise property of the nanomaterials which can take off in further applications. So when we consider about the nanomaterials and its theranostic applications especially, then three things of course we need to consider. I am showing you a virtual picture about a theranostic nanoconstruct. You can see it. It has major three components. One, its biomedical payload. One as a carrier, and another is the surface modifications. Payload is nothing but the molecules of interest which having therapeutic potential. It's the drugs or an imaging agent. I can discuss in the coming slides more. And the carrier is a nanomaterials which have different size and shapes, you can see. And the surface modifiers, generally surface modifiers are used in different ways, using some biocompatible materials, using some targeting moieties as a surface modifiers. And as you see, as I said, the biological payloads are all drugs, therapeutic agents, and any imaging agents. So these are the very basic concept of when you think about or imagine about a theranostic construct. So coming to the cargo, as I said, the therapeutic molecules, then these theranostic nanoparticles, when you have a cargo molecules, which is a therapeutic molecules, it has two features, or it has two kinds. One, we can use the imaging agents, and another we can use the therapeutic agents. So you can see again a virtual cartoon where we can have a therapeutic molecule as well as an imaging agent. So both are in a synchronized fashion, which can enable you to give a very crucial, focused, targeted medicine, and that medicine, when it's given to a system or living system or disease system, then that therapeutic progression can also be monitored by this imaging agent. So now you can consider that it is a combination of the therapy and the diagnosis coming to play in a synchronized fashion. So coming to our, as I said, the imaging agents. So these are the so-called imaging agents are now practiced clinically. There are multi kind of optical imaging systems having MRI, PET, ultrasound, and the CT imagings. All these imaging modalities having different kind of features. You can consider it as a pros and cons, and that can be tuned which one you need it for a particular treatment or particular therapeutic potential to gain it. So now coming to the, as my talk on the cancer theranostics. So we need to think about what are the therapeutic agents which is focused for the cancer. Actually, there are 10 hallmarks of cancer which are considered today that gives a insight to think about how we can impose our therapeutic molecules. So in this kind of pie chart, you can see that there are 10 features for the hallmark of cancer, which can raise a cancer. And to fight on that, we have a therapeutic modalities, which may be a small molecular chemodrugs, targeted antibodies, which can be used as a targeted therapy, also non-viral or viral gene therapy, hormonal therapy, and any other therapies which I'm going to discuss again. Now coming to the carrier, as I said, the carrier is nothing but a nanoparticle. It is a vehicle that can ferret the cargo molecules towards the target site. So there are many kinds of carriers you can imagine or you can think about to construct, which may be a biocompatible polymers, which can be a lipid, dendrons, or any kind of inorganic construct molecules. So this here, I can show you how we can build up the carriers. Suppose it is a polymer lipid bilayer carriers, where you can see it's like the core cell nanoparticles, and it is another kind of polymatrix. Some of the carriers having a balance of the hydrophilic and hydrophobic core and cell units, which can enable to carry the drugs which is suitable to the target site. So as per the nature of the drugs, whether it is hydrophilic or hydrophobic natures, we can make these carriers suitable so that we can adopt or load maximum number of carriers. So just I'm going to give you the features, and when the nanoparticles or particle carriers we are considering, then we also need to have the things about what kind of barriers which need to overcome to deliver a nanotheranostic moieties. So if you consider the barriers, or for that what we need to overcome is that the carriers should be very good in the circulatory system, means high solubility in the blood, and it has to overcome the RES, reticuloendothelium systems, and it has to optimize the size range, and it's having huge amount of biocompatibility and minimal toxicity. These are the very good physicochemical features we need to adopt when we are considering or making a construct on that. So coming to that, again, the barriers. Basically, there are two kind of, or broadly classified two kind of barriers. One is called the cellular barriers. Cellular barriers are the cell membranes, or in the intercellular membranes like the organelles like endosome, lysosome, they are membranes which we have to traffic on that. And another is the physical or physiological barriers, that is like blood, liver, spleen, all these organs which we can need to overcome to penetrate on it. So thinking about to build up a nano construct, you have to think while we are considering, when we are applying this in a physiological systems, we have to overcome either cellular barriers or is a physiological barriers. So considering these two, we can say it is a biological barriers, and these biological barriers, what we are going to make it, that slide is giving you another very good insight. So if you can see, when you consider, as I said, the physical barriers encountered by the nanocarriers or nanoparticles, these are the features. It has a like blood-brain barriers, and that would need to penetrate the transcellular lipophilic pathway, where we can have to ferret the nanoparticles in the following pathway. And another is the adsorbate transcytosis pathway. There we can have a phenomena, it is called the adsorbivity, where we can ferret the cargo molecules based on their charge imbalance or charge balance. And another is called the receptor mediated. And we can also having the pathway, paracellular aqueous pathway. So considering the right side, if you can see the cellular barriers where you need to overcome, then you can think about there are few features or phenomena. When a carrier comes to near to the cell surface, and then we have to see the charges. You can see the charge between this cell surface and your constructed nanocarriers. Then when this charge, it is a electrostatic fashion, then it called the adsorptive pathway. Well, it can be entered through this pathway. Then another is the direct penetration also happening in some cases. And most important features is the receptor mediated pathways. In the cell surface, lot of receptors are there. If you make a carrier which can recognize that kind of receptors, then it will recognize first the receptors in the cell surface, then it can internalized by these receptor mediated pathways. And these receptor mediated pathways are different kinds. One, we can say it is a endocytosis, and another we can say it is a phagocytosis. So again, it is classified different kinds: macropinocytosis, Clavelock or Clathrin mediated endocytosis. So these are the small features determined how it goes to the cells. The main aim is that it should enter to the cells with this nanoparticle and then release the cargo at the intercellular targeted areas, either it is organelles or in the intercellular systems. So for that, we have to consider how we can feature out our nanoparticles to overcome the cellular and the physiological barriers. So currently, as we are today, I'm going to talk about the cancer theranostics. So we, as a general phenomena, what are the cancer therapies opted today, or we are using? Starting from the chemotherapy, we can have different kinds of left and right side. These are the therapeutic modalities are practiced in the clinic. But all these therapies are very subjective, and we have to consider which therapies we can adopt as a nanoparticle or nanocarrier development, and that can give the maximum therapeutic index. So starting from that, I just give you some of the examples. As the first thing is the chemotherapy, what I consider, which is an anti-cancer drug. So what are the anti-cancer drugs? Is a general questions, and what its features. Actually, I'm going to say you there is a broadly classified 6 types of the anti-cancer drugs, if you consider, and which can use for a cancer therapy. It should be alkylating agents, and it should be antimetabolites, anti-tumors, and it can having some kind of specific hormone targeted therapy, and there are different kinds of other miscellaneous cancer drugs. So these are the general features to consider which kind of cancer drugs and how we can monitor. In the area of the drug discovery, they are focusing the small molecular drugs based on the general features, and which that general features are also adapted by this kind of classifications. So coming to another kind of therapy, which is also now evolved, it is called the phototherapy. So phototherapy is a kind of therapy where we can illuminate light, and that light can generate heat, and that heat can kill the cancer cells. This is very simple way I'm just going to tell. So this light, either we can generate the heat through light, or we can generate heat through magnetic field. So when we generate through magnetic field, it's called the hyperthermia, and this heat having some specific features, because how much temperature you need to generate to kill the cancer cells, how it is localized to the cancer cells, that all feature out using 2 modalities of the generation of heat. One called the photothermal therapy, and here in that kind of therapy, you need a light source, and also you need a nanomaterials which can activate and generate the heat in presence of light. So there are 2 kinds of light source generally used for the PDT in the NIR1 and NIR2 windows, which can generally illuminate the light and then generate the heat. So when nanoparticles falls on the capture these irradiations, and due to some of the plasmonic nanoparticles which can generate the plasmon imbalance, and then can having some electromagnetic changes which can raise the temperature as well. Another is called the photodynamic therapy, where you need to have a molecules which is called the photosensitizer. It is a organic molecules, or it is a complex metal ligand complex, may be possible. But the feature of these molecules that on tuning the light, it can generate the singlet oxygens, and that singlet oxygen is a toxic materials which can damage the cells, damage or intercalate the DNAs. So it is called another phototherapy, the photodynamic therapy. these are the two therapies now practiced slowly in the clinics, and it is now in the hot area of the cancer research. Apart from that, we all know the conventional radiotherapy. I'm not going to discuss much about the conventional radiotherapy, but you can see that the conventional radiotherapy are utilized in the following types of cancer treatment. And now coming to the era of the immunotherapy. So immunotherapy, apart from colloquial the cancer therapy, if you see the immunotherapy, which we can activate our immune systems. So how do you do that? You know that if you have activate our immune systems and which can generate the activate immune cells. So immune cells generally in terms of as a usual way it is inactive. So one of the immune cells, just I can say that T cells, which if you can activate, so activated T cells, this activated T cells in terms of its activations can proliferate the cytotoxic T cells, and these cytotoxic T cells can kill the tumor cells. This is very general way I'm trying to understand that in a immune activations, apart from the chemotherapy, it can be used to complete eradication of the tumors. Now you have all heard about the immune checkpoint therapy. It is nothing but there is a checkpoint between these immune systems, and if we can block this checkpoint, then we can easily activate these cytotoxic T cells, and which can generate the antigenic part and also kill the tumor cells. So I'm going to talk about the kinds of cancer therapy which is evolved. Now people are not trying to use in a fantastic way the multimodal therapy, using the combination of two kinds of phototherapy or a choice of photochemotherapy and the chemoimmunotherapy. And these kinds of therapies are now very much practiced in a research mode. Also, they are going to translate in the translational mode. So I'm talking about all these modalities in last 10 years advancement. So I'm coming to in the end that these therapeutic modalities in the research mode or in the clinical translation, how far we are now. So far I talked about the therapeutic modalities. Now coming to the diagnostics on the modalities in the nanotheranostics. So as the diagnosis or diagnostics is one of the integral part of the nanotheranostics, so we have to consider what are the diagnostic features or the diagnostic modalities. We know there are several kinds of imaging techniques are used in the preclinical and the clinical research, and all we know about this one like CT, MRI, PET, ultrasound, all this. So if you consider first the optical imaging, so optical imaging, first we need to think about the fluorescent probes, which is widely used in the imaging in the cellular systems and in the organs and the tissues. And most of the popular imaging techniques also use like imaging contrast agents, and also we are using different kinds of NIR or fluorescent probes, which can use for the seeing the deep tissues and to image it. And also now the concept is that targeted imaging. So imaging molecules, if we have a targeting groups, we can use to see where is your localized dysplasia is. So these are the optical imaging modalities. Now coming to the MRI, and all we know, I'm not going to say about the details, but I'm going to say that this is one of the modalities are used in the practice or clinical use, mostly certain kinds of the diseases apart from the cancer also. And they have used kind of contrast agents, and these are contrast agents they are using for this MRI, and measuring the elasticity of different angles like T1 and T2s, and we will get some of the features of the image of the MRI which can recognize the diseased area very easily. And another is the photoacoustic imaging. and where we can see it is a also one kind of imaging, along with we are using the CT and the MRI. So these are the imaging techniques, although it is a very expensive, but it can be utilized. And to get a particular therapy, especially in the cancer diagnosis, these are the imaging techniques are now being used in the clinics. And also this is called the positron emission therapy. This is also used in the clinics. And this will also giving a particular localized area where the cancer cells and or tissues are located. So it gives a very tremendous potentials in the in vivo part of the tumor and its biodistributions as well as its pharmacokinetics in a non-invasive manner. As I said, the CT also one kind of such techniques which is also used in the clinics nowadays. And again, I'm telling you, the changes or revolution is coming, that two imaging techniques coming together to reconfirm or using as a complementary techniques like PET CT, PET fluorescence, and PSCT and MRI. So these are the usings, and these are the classifications of the cancer nanotheranostics. As I said, that in the features you can see the lipid-based carriers and polymer-based carriers, inorganic materials as carriers, viral nanoparticles, and the drug conjugations. So if you look on these whole things, you can consider that if we want to make a nanotheranostics, then you have to consider all this. And first you have to look which type of cancer treatment is you need to work on and which kind of carrier is suitable on that. So that combination and choice is very crucial in this research area. So that's why I'm going to tell you, and it is like a 2x2 features. Suppose in the columns I'm telling you all the optical imaging and units, and in the here rows I'm giving you all the therapeutic modalities. So you can choose which one you can choose and which one will give the complement, giving the maximum theranostic potentials. And among this, imaginations and thinking will come that this may be a potential for this particular therapy. These are the kind of nanomaterials. All of you know that gold also used widely as a nanocarriers because of its excellent biocompatibility, and it has the surface modifications, easiness of these surface modifications, and it has, we can make or tailor-made different kinds of cells. Like that, this is the mesoporous silica nanoparticles, which is also featured as a good nanocarrier or nanotheranostics systems, where we can use different kinds of shapes. And most important thing is that mesoporous nanoparticles is that it has different size or size tunability pore size, where we can load the drug or cargo molecules on it. And these are the carbon nanoparticles. Also, people are working on the different kinds of carbon nanoparticles as a good amount of carrier development. And another features, now you can see in the recent last 5 to 10 years, upconversion nanoparticles, which is widely used. Upconversion nanoparticles are mostly the lanthanide groups or lanthanide dopes particles, which having a sequential absorption of multiple photons in the lower energy that can produce in the higher energy anti-stock luminescence. Actually, from that, if you can use a higher energy sources, then we can see or we can modulate the imaging features in the either in the visible range or in the UV range. And another term, I think all of you heard about the aptamers. Aptamers, nothing but a small fragment of a oligonucleotide, and it has a definite targeting motif which can be utilized as a targeting groups. Suppose all of you know there's antibodies. Antibodies is a big molecule. It has the molecular weight more than 100 or nearly 200 kilodalton. And if you, and also if you can see a big amount of oligos or DNAs, which is having very high number of base pairs. So if you consider a very core targeting unit having 10 to 20 base pairs of oligonucleotides, which is called the aptamers, which can be used as a nanomedicine targeting motifs. Another term recently comes around 5-6 years, which is called the exosomes. Exosomes are the extracellular vesicle particles, which is coming out the extracellular vesicles and originated from the cellular environments, and which can carry the genetic material, as well as it has a very good potency of the targetability. So now the area of the exosome research as a exosome as a nanocarriers, we can generate a nanocarrier from a biological systems because of its easiness to deliver the carrier and easiness to make as a targeting motif. And there are so many of the polymeric carriers. So whatever the general features I'm going to talk about, now I'm going to some of the giving of the examples and the realistic features what our people are practicing. These are the polymeric nanocarriers which our people are using, and there are quite number of supramolecular nanocarriers are also used based on the one of the strategy that they can generate the fluorescence using different kinds of strategy. One is the aggregation induced emission strategy that can be adopted through this using these supramolecular features. And another is the viral-like particles. People are also using, as I said that earlier, the non-viral gene therapy. So the viral-like particles can be used as a carrier molecules. So these are all about what so far I talk about all the general features of the theranostics, concept of the theranostics, and what kind of carriers we are using. Now I'm going to give you another 10-15 minutes. Can I get another 10-15 minutes?
You can continue till 12:10. That is the 45 minutes.
Sure. Thank you so much. So now I'm going to give you some of the general features and what we have developed in our laboratory so far. So as I said, in the theranostic dimensions, theranostics is a combination of diagnosis and therapy, two modalities together. So if you consider the therapeutic modality, two are the phototherapy, one chemotherapy, and on radiotherapy we can considering. And if you see the diagnostic modalities, I can tell about this is the fluorescence. We know the MRI, and I also give you the briefness about the computed tomography, CT or PET. So all of you probably heard about the Raman scattering. So today, one of the features of the Raman scattering, as I can also give you in the subsequent slides, the Raman scattering is also used as a diagnostic modality in biomedical applications. And one of the modified versions of the Raman scattering is called the surface enhanced Raman scattering, which is in abbreviated is called the ACRS. So this having huge feature and huge importance in the diagnostic modality. I'll give you some of the insights of the SIRS as the diagnostic modality. And these are the combinations why this kind of modalities are used, as I already discussed earlier. So coming to the first, I give you some of the true examples or the ongoing work on the molecular diagnostics, and which based on the definitely nanotechnology. So if you consider the enrichment detection, so there is, I want to focus you about the surface enhanced Raman scattering as one of the diagnostic modality, which is now evolved one of the major breakthroughs in biomedical research. So if you see that in biomedical applications, especially the clinical detection or diagnosis, there are two kinds I can describe you in the later slides, where level-free and level-based detections, which can be used, and monitoring the cancer therapies, and also monitoring the surgical performance and the biosensings using these modalities. So as you know, what is Raman scattering? All of you know it is a inelastic scattered lights, when a small fraction of the light which can be having different energies compared to the incident photons is called the Raman scattering or Raman effect. But the thing is that this Raman scattering is very weak phenomenon. So for that, actually 1 by 1 millionth of the scattered phone coming as a Raman photons. So for that, there is a phenomena where if you embed a nanoraphane surface and put your analyte of interest on it and put on the lasers, then you can see the Raman fingerprint of this particular analyte, which can be enhanced up to 10 to the power 6 to 14 fold. And there are two mechanisms behind it. One is called the electromagnetic enhancement and the chemical enhancement. So this features is called the surface enhanced Raman scattering. For that, the basic need is that you have create a nanoraphane or metallic nanomaterial surface, or as a such substrate, which is called. So there are a lot of kinds of such substrate are there. The basic such substrates are the gold or silver nanoparticles, or it's a chip-based nanoparticles which can use to enhance for the analytes. As I said, that level-free or level-based Raman spectroscopy. So this level-free search means you have the nanoparticles, which is a search enhancer. So if you use these nanoparticles directly to the analyte of interest, then this analyte come and form in close proximity with these nanoparticles in the surface, and it giving a enhanced biomolecular fingerprint of these analytes. It is called the level free. Whereas the level based, you have a suitable Raman reporter which can give a specific Raman signature peak that can be incorporated in the nanoparticles as well as your targeting groups. So when this targeting groups, like in terms of antibodies, if you conjugate, then it is called the Raman tags. And when these Raman tags are incorporated in your analyte of interest, then this recognition happen between the antigen and antibodies. And when this recognition happens, you can see the Raman signal of the reporter molecules that can give you the recognition modality. So this is called the Raman tags or the SARS tags. So there are a lot of examples in the biosensor development in the diagnostics fields, and which can complement with the complementary features on that. So I'm going to give you, as the cancer is one of the major topic here, so I'm going to give you the features of this recent advances in the diagnosis in the Raman scattering. So why it is important? Suppose all of you know that the cancer cells and how we will kill the cancer cells. There are two ways that is happening. One is called the apoptosis and another is called the necrosis. So when we can generate a programmed cell death, apoptosis, there are the features happening. One is called the protein denaturations, protein degradation, and DNA fragmentations. And these minute biomolecular changes, suppose a protein denaturations. Protein having two states, one is the native state and it's had a denatured states. In the native states, it has lot of disulfide bonds which can accumulate the proteins, these SS bonds. But when it is the denatured states, these SS bonds are cleaved. So through Raman fingerprint, we can monitor the cleavage of these disulfide bonds. And so protein denaturations can be monitored here. I'm just telling you, you can see the major peaks here, around 838 peaks, which is very much prevalent in the denatured states, but it is not coming in the nature states. And some of the peaks, this 1000 peak, which is very much prevalent and in the denatured states, but not in the native state. Because the denatured states, the proteins are segregated. And here, these 1,000 peaks are resembled the phenylalanine. So there are a lot of amino acids and protein fragments can be identified by these techniques. So I'll give you more examples. And there are works coming on last 5 years. I'm just going to give you the briefness of that work. Suppose the gastric cancers, there are 2 stages. One is the early stage gastric cancer, in the advanced stage. So people are working with the nanomaterials. We know that graphene or graphene oxide, that is a SIRS enhancer. So if you put on your breathe on that substrate, and then you can accumulate the Raman fingerprint of the biomolecules present in your breath, and which can be enhanced by this graphene oxide. And this Raman fingerprint further classified using a chemometry, where you can classify these 2 classes of the major stages of these 2 classes of these gastric cancers. And there are examples where in the surgical resections, where you can use that where is your exactly located the cancer area and the normal area using this SIRS modality. So I'm not going to give you the brief explanations, but I'm just showing you how it can be features. As I said, that apoptosis and necrosis, which can be easily distinguished by the fluorescence using the FACS analysis. Suppose here you can see the cells which is coming as a viable cells, and is the apoptotic cells and the necrotic cells. That can be easily differentiated by the FACS analysis, all we know. But that can also be differentiated by the plasmon enhanced Raman scattering. So you can see the Raman spectra, all these 3 classes, you can see the difference in the Raman's fingerprint that can be classified and also easily tracked by these features. Coming to our own work, last couple of years we are working on the cervical cancer detections. You know, the cervical cancer generally detected through Pap smear test in the cytopathology. So what we plan to do, that this work is done in collaboration with the Regional Cancer Center, Sivanandan, where actually this cervical cancer cells, which is collected from the uterine cervix, having 3 general major features. You can see in the normal features and the intermediate and the the carcinoma features, which is fully carcinoma stage. What we did, we have collected as a group of all these cells, and these cells we have tried in 3 ways: as a single cell palette, and their isolated DNA. So all 3 sets we have tried to evaluate the Raman fingerprint, and this Raman fingerprint further through artificial intelligence platform, through chemometry, we can have a very good group or separation modality, where we can separate these 3 classification very easily. And that features are now complemented with the usual cytopathology techniques. So we have now a sufficient number of we have done, and the validation still is going on this platform, but these Raman techniques can be easily grouped. And here the cervical cancer in another way, through level-based, we have also classified in the single cell analysis. Similarly, in the classical way, we have also worked on the lung cancer, and here the lung cancer we also determined using these features. Actually, there are quite 3 major biomarkers we have tagged, and 3 major biomarkers in a complementary method in a single platform as a multiplexing way we have identified in the lung cancer, which also validated in the cytopathology techniques. Also, recently we worked on the breast cancer. Generally, there are 3 major important biomarkers, which is called the prognostic markers: hormone receptors, ER, PR, and HER2, which can be tracked and determined in a single platform what is the presence or absence of these 3 biomarkers. Because these are very important things. Based on these features of these biomarkers prevalence, the treatment modality is determined. But the key challenges for the oncologist, that they are very much confused by this cytopathology technique, in a single slide which biomarkers is there. If ERPR is present, then the treatment modality is different. If HER2 is present, this treatment modality is different. If all three are absent, which is called the triple negative breast cancer, then treatment modality is quite different. So through Raman modality, we have developed a strategic plan in a multiplex platform where we can differentiate three kind of biomarkers in a single tissue samples. Also, we have worked on the prostate cancer, where we also differentiated or identify the presence of the PSA level in cellular system we have done, and we have identified the PSA level, and which gives a very good detection limit. So these are coming about the diagnostic modality. Now I'm going to give you another 2-3 minutes, going to finish with the therapeutic modalities. And suppose we want to give a theranostic ideal on this one of the feature molecules. You know, this molecule is called the doxorubicin, one of the anti-tumor drugs, which is conjugated through a gold nanoparticles, and it has the linkage and the which can be clipped. This oxime linkage can be clipped in the cellular system in the acidic pH. So when it is clipped, then doxo will be liberated from the gold nanoparticles, and once doxo is liberated, it has inherent fluorescence in the more or less in the red regions. We can see the fluorescence turn on, and when it is apart from the nanoparticles, then its surge signal going to come down. So it is a turn off on mechanisms are established as a nanotheranostic platform. So we have also worked on the nanotheraноstics using gold nanoparticles, and also we have used this doxo, and we have also clipped and these mechanisms, and we also delivered this platform here. So, and also therapeutic potential also be classified. And one of the important features here that when in the molecular level, when this doxo can create its therapeutic potential, it is used as a DNA intercalating agents. So when it's clipped, then these Raman features can be easily tracked. Suppose you can consider this 838 peaks, which is not present when it is clipped or when it is fully apoptotic conditions. There are few targeted nanotheranostic agents we have also concluded here and also published is the Mesophora silica core cell nanoparticles, which also used using a dox-loaded nanocarriers, and we have using the folate targeting nanoparticle using the folic acid as a targeting modules. So my time is going to finish, so I just skip some of the slides. And what I want to say that these nanotheranostics, especially in the cancer modalities, are coming up in the preclinical and the clinical features where we can do. We also work in the melanoma, and also we have using this FDA-approved drugs, decarbazine, and we also deliver in this melanoma, which can be monitored both Raman and as well as in the fluorescence modality. And this is very recent work. We have utilized DOX and the drug efflux inhibitor Tericuda. So both can be used in a single construct to complete eradication of the tumors. And that also we have recently developed and also utilized in the animal models. And these are the very recent features in this year published. It is a nano engine where using Mesophora silica nanoparticles and as well as a periodic Mesophora organosilica as a nanorod. So nanoparticles and nanorod combined together, giving a good therapeutic modality and giving high loading capacity of these dogs. So this is also a very recent work, and that I am going to skip because probably my time is going to over. And these are very recent works which is accomplished using both chemo, photo, and immunotherapy features. But this is probably my last slide. What I am going to say about the success stories of these cancer nanotheranostics. As you see here, there are a lot of drugs which is named in their trade name, which are clinically approved and which are used in the clinics as a outcome of this cancer nanotherapy or nanodelivery systems and nanotheranostics. So one of the drugs I can emphasize here is Doxil, which is liposomal dox, which is used and which is developed by our Indian company Sun Pharma, and which is now FDA approved and is clinically used. So we are looking forward more such kind of drugs or nanotherapy or personalized medicine will come. So these are some of the futuristic perspectives in these areas. And probably in the last slide, I'm going to tell you what is the market potential in this research. So people are expecting, and statistics are telling, that molecular diagnostics will come up around 13.8 billion in 2025. And cancer nanomedicines, which is a booming area, which also coming hugely, around 387 billions by 2027. So I want to say, take a message to all the students, faculty, or students community, that it has a potential in huge in the area of nanomedicines and nanotheranostics and personalized medicine if we work very clever way to build up these systems. And also it requires equal importance, not only it is a multi-domain area, chemist, biologist, material scientist, and also clinicians are required to come up with this venture successfully. So I like to acknowledge all the peoples. With them I am working, and with their support I am able to present you. And this is my group. My group is a consist of biology and the chemistry team, and they are solely working hard. And for them, I am going to be able to present here the features here. And thank you all for your kind answers. Thank you.
Thank you, Dr. Kaustabh Kumar Maiti, for your very fascinating and illuminating lecture. For the last 45 minutes, we were so much enjoying the various tenets of nanoscience, nanomedicine, and nanotheranostic research propagation happening in the field of material science and medicine. Your outstanding contribution is mostly appreciated in this international conference. Now this presentation is open for discussion. The participants can ask a few questions. Please.
Sure.
Dear delegates, join for some discussions. Then only the presentation will be more active and very positive and productive. So one question is here from Cheryl Cherian Parakal.
Yeah.
Dear sir, the nanocarriers, nanoparticles may cross the blood-brain barrier and accumulate in the brain, possibly causing adverse neurological effects. How can we modify them to prevent them from crossing the barrier in the cases where it is needed?
Yeah, it is probably a good question, and that also is a challenging one. So tuning the nanoparticles to cross the blood-brain barrier is a challenge, definitely. So what people are doing, that the nanocarriers, as I mentioned, the size is very important, and to tune its lipophilicity and hydrophobicity, that also be tuned in a way that it can cross the barriers. And for that, most important thing is that we have to have a biocompatible polymeric carriers, which is shown some extent it can cross the BBB, and it has the size range. If you can come out within 50 to 80 nanometer size, then it can definitely cross with the tunability in the polymeric materials to enhance the biocompatibility. So the feature is that if you can tune the size within this range, and it has great scope to cross the BBB.
The next question is from Rincha CH. What about the effect of serum albumins on these nanoparticles once it entered into the body?
It is difficult to say. This serum albumin nanoparticles may interact with the serum albumin, but in some cases it can have some adverse effects surely. But in most of the cases, as the nanoparticles what is used in the nanotheranostics are very much biocompatible and also biodegradable, so it may not have much adverse effect on the serum albumin.
Thank you, sir. Any more questions from the audience, from the delegates and students? Sir, I have some suggestions and clarification from you.
Sure.
And most of the research nowadays in the chemistry is on material science, particularly on nanomaterials. Your research is very, very important, and ultimately it reach to the public in the form of a medicine or a diagnostics tool. The component of chemistry is huge, and analytical chemistry is also there. In a research institute like NIST, it is possible because multidisciplinary level of research and departments are there. From the university departments, how the students can indulge in this kind of multidisciplinary and socially and having much more relevance subject like you delivered here, how that kind of research can be initiated from the limitation of a university research facilities? Because a lot of people are involved, the medicinal people and medical practitioners, their support, then analytical instrumentation, even the Raman spectroscopy, a lot of participations are involved. In that respect, what advice you can give and can share with the students community now?
This is a very good thinking, sir. One way I can feel now that for the university setup like CUSAT, I know they are also slowly very kind of motivating the students, and also their also culture. The basic thing is required a multidisciplinary culture and also aggressiveness of all the faculties to have a joint thinking and joint project which having these potentials. So I surely think that you have one kind of centers like inter-university, some centers I have heard about. So if the individual departments and centers can think about some joint program and which they have contributed their own capacity or own expertise and make such kind of multidisciplinary work, like suppose you are on to make a delivery systems or nano delivery systems, and which can be reached some extent to the clinical or preclinical level. So basic things that a joint collaborations within the university system first, because I believe that charity begins at home. So first you have to try make it in your own system first. Definitely it is not enough, I'm telling you. Requires that then next step should be the nearby institutes and nearby centers where they are most willing to take up such kind of collaborations. That's what I try to make it, because even in the CSIR-NIIST, I'm telling it is not at all enough. So here I find that there are institutes like RGCB and also the RCC. There are people that there are faculties that they are also willing to work together. So most important things that we have to more aggressive to make these collaborations. Then only it is possible.
Thank you, sir. You are an excellent motivator and a teacher. Your lecture unfolded with the basic concepts of nanomaterials, what is a nanoparticle and how it can be implemented for diagnostic determinations, and the molecular biology aspects you discussed, how new drug formulations can be made for diagnosis. And every time the basics and the fundamentals of the applications you discussed in a very simple language. And ultimately, it ended with your own experience and your own research contributions to the society, with the concept of nano engine and the success story, and elaborately discussed how Raman scattering can be utilized for this kind of diagnosis in our level of diagnosis with respect to the determination of the gastric cancer using nano gold materials, then cervical cancer diagnosis, then prostate cancer diagnosis. These terms are very, very annoying, and a lot of fear is induced in the heart, in the mind of each and every Keralites, because cancer is a great concern, and every people have always have a bad feeling about what is going to happen the next period of time. So in that respect, your contributions are more humanistic, and ultimately it end up with many success stories with respect to the innovations with respect to delivery systems, and also you concluded your presentation with the market share and the future prospects, how the students can indulge in this very important cutting edge research area where multi-facets of research is involved. On behalf of the organizers and the delegates and the participants, I extend you, sir, our heartfelt indebtedness for giving a very illuminating lecture in this very important MATCON 2021. Thank you, sir. Thank you very much.
Thank you. Thank you, Arun. Thank you.
Now we will continue with the remaining our presentations. We have Dr. V. Sivanandan.